amazing AP Bio 2 kiddos, it's Miss Lynham, and today's lecture is going to cover carbohydrates. Okay, so let's go ahead and get started. The first thing that I want you to notice on the bullet, these are functions. Okay, so when you're studying, these are functions of carbohydrates. The first thing it says is provide fuel. Okay, so there's two things you want to think of with the fuel component, and that is carbohydrates can be broken down into sugars and starches. Okay, so the sugars, the first word, is going to be immediate energy. So if you eat like a sweet tart or some Skittles or some type of sugary thing, the sugars are going to provide you with a quick energy source, whereas if you eat a starch, which is also a carbohydrate, that is going to be more stored energy. That's how I abbreviate energy. Okay, so sugars and starches are the two groups of carbohydrates, so that's one of the functions, is to provide us with fuel. Okay, another function is to act as a building material. Okay, we're going to talk about cell walls, specifically of plants, and then also of fungi, and how those cell walls have a material that is a carbohydrate material that helps to give that structure and support. Okay, also we're going to talk about the exoskeleton of arthropods and insects, which has a carbohydrate polysaccharide in it that also gives it a certain function. Okay, one that's not listed here that we did talk about freshman year when I had you guys in Bio 1 was carbohydrates can serve as carbohydrate ID tags. Okay, meaning that they can also attach to protein molecules to serve as identifiers for cell types. Like it can identify a cell as a skin cell, or as a white blood cell, or cardiac muscle cell. So a carbohydrate can attach to a protein and become something that, if you read in the book, it was called an oligosaccharide. Okay, an oligosaccharide, which is a type of carbohydrate that attaches to that protein to serve the function as being an ID tag. So carbohydrates have a variety of functions. Generally speaking, when you look at a carbohydrate, the formula is written as CH2O. And that's kind of where the name comes from. If you look at the name carbohydrate, you guys know that hydrate means water. Carbon refers to carbon compounds. And that's because if you reduce the formula, you get carbon plus water. That's kind of the reduction of that. I mean, really, honestly, you really don't get carbon and water because we have the carbonyl functional group and the hydroxyl groups. But you can take every carbohydrate and reduce it, and you should get a 1 to 2 to 1 ratio. For example, if I gave you the formula for glucose, it's C6H12O6. If I reduce these three to the common denominator, 6 goes into 6 one time, 6 goes into 12 two times, 6 goes into 6 one time, I get 1 carbon for every 2 hydrogens for every 1 oxygen. That's the same thing as what you see written here as CH2O. Okay, so that's the general formula. So you're only going to see carbon, hydrogen, and an oxygen in a carbohydrate with a ratio of 1 carbon to 2 hydrogens to 1 oxygen. Okay, the third bullet describes the functional groups that are present in a carbohydrate. Okay, so I introduced a couple of days ago the carbonyl, okay, or carbonyl functional group. This is the carbon, okay, let's see if I can get this right, the carbon that is double bonded to an oxygen. Okay, that's the carbonyl functional group. Plus, carbohydrates are going to have multiple hydroxyl groups. Okay, the hydroxyl group is the OH group. Okay, so that's the OH group. So a carbohydrate is going to have multiples of the hydroxyl groups, and it's definitely going to have a carbonyl group. Okay, so if you look at the next bullet, it's talking about the monomer. Okay, so let's revisit that term because I introduced it to you at the beginning of this biochemistry unit. Mono, the prefix mono means one, and then don't forget mer means unit. Okay, so this is 1 unit. It's the building block. It's the simple building block that makes up a carbohydrate. So the monomer, the building block, the 1 block of a carbohydrate, is known as a monosaccharide. Once again, if you use those Latin roots, guys, then you can take saccharide, S-A-C-C-H-A-R-I-D-E, and that translates into sugar. That's what saccharide means. Okay, so if I take a 1 sugar, I get a monosaccharide. That's my simple sugar. Okay, so in the next bullet, there are different examples of monosaccharides. Okay, they're usually found as ring structures, which is what you see down below. A ring structure means when it's put in an aqueous solution, it tends to take the shape in a ring. Okay, and the ring can be either a pentagon or a hexagon, depending on if it's a 5-carbon sugar or a 6-carbon sugar. So the first bullet here, you can see these are called pentoses. Okay, so anytime I have OSE or saccharide, S-A-C-C-H-A-R-I-D-E, both of those refer to sugar. So use your Latin words. If I put pentose, I get 5 sugar, it's a 5-carbon sugar. If I put hexose, okay, that's a 6-carbon sugar. So we can have 5 carbon sugars, we can have 6 carbon sugars because they end in OSE, that's the tie that means sugar. Okay, so my monosaccharides, my single building blocks, my 1 sugars, are either 5 carbons big or they're 6 carbons big. Okay, so look at the examples that are listed here for the pentoses. These are the 5-carbon monosaccharides. We've got ribose and we have deoxyribose. Okay, we're going to elaborate on these more when we get to the nucleic acids, but if you look at the picture, okay, you can see that these are definitely in the shape of a pentagon, so those are the 5 carbon sugars. The one on the left is ribose, the one on the right is deoxyribose. This one's found in DNA, and this one is found in RNA. Now, don't confuse the biomolecule group, which is called nucleic acids, because a nucleic acid is another group which DNA and RNA are examples of. But DNA and RNA have a sugar component to them, which are these 2 sugars that are listed here. Now, if you're wondering what the difference is between these 2 sugars, I was hoping you guys would recognize, it has to do, okay, with the number 2 carbon. So let me show you a couple of rules in organic chemistry. On the pentagon, this is the number 1 carbon position. There's a carbon there. This is the number 2 carbon, the number 3 carbon, 4 carbon is here, and this carbon at the top is the number 5 carbon, the one that you see right here. Okay, so that's the 5. So if I look at the number 2 carbon slot right in here, in ribose, I have a hydroxyl group right here, OH, attached to my number 2 carbon. And look at the word D, DE means without, oxy refers to oxygen. Look at this number 2 carbon for the sugar in deoxyribose. So at the number 2 carbon slot, it is missing, that's the DE, without, there's no oxygen right here. Okay, that's what makes deoxyribose and ribose different from each other, is the fact that deoxyribose is missing an oxygen on the number 2 carbon spot. Okay, so that's just showing you some similarities and some differences because they're 2 totally different sugars. Okay? Alright, now let's visit the hexoses. These are actually the more common ones that you are familiar with as far as eating and foods. Okay, so these are hexoses, which are 6 carbon sugars, and the 3 that you see listed here, glucose, fructose, and galactose, all have the formula C6H12O6. All 3 of those have that same chemical formula. But if you look at the picture to the right, okay, let me scroll this down a little bit. These are the linear structures right here. This is the linear structure of glucose, linear structure of fructose, and linear structure of galactose. If you notice these 3, there are some significant differences. Here's the number 1 carbon in the linear structure. Okay, here's the number 1 carbon of the second one, number 1 carbon of the third one. If you look at the number 1 carbon in glucose, the carbonyl group is located on the terminal carbon. Look at fructose, the second one. The carbonyl group is found within the carbon chain. And then over here for galactose, it's back up to the terminal carbon. So glucose, the first one, and galactose both have terminal carbonyl groups, whereas fructose is embedded in the skeleton. Okay, that is definitely a structural difference. Okay, if I compare now my glucose, this one on the left, to my galactose, if you look right in here at the number 1, 2, 3, 4th carbon, okay, so right in here, the number 4th carbon, if I can get my pen to work, right in here, number 4 carbon has a slight shift in the OH, which is the hydroxyl group. Okay, so look over here. On the galactose, the OH group is on the left-hand side, and on glucose, the OH group is on the right-hand side. So that one difference makes those 2 structurally different as well. Okay, so let's go back and revisit what we just said. Glucose, fructose, and galactose have the same chemical formula, okay, but they have a different structural arrangement, okay, different structure. What we call that, there's a name for this vocabulary term, these are known as isomers to each other. Okay, in chemistry, an isomer is anything that has the same chemical formula but a different structural arrangement, and that's what you see in this case. Okay, now as far as really in reality and biology, how are they different? I can tell you glucose is the preferred. Glucose is the one that the body prefers. That's the one that supplies cellular respiration, the mitochondria. That's what supplies that process with that energy source. Glucose is the one we prefer. Okay, glucose, also, the preferred one, I'm going to bring a line down here, that's the one that plants make in photosynthesis. Okay, so that one's made by plants. Fructose, the second one, this is the sugar found in fruits. As the name implies, fructose is fructose, or it's a sugar that is made by fruits of plants. It's very sweet. Okay, that's a highly sweet one. Glucose is not as sweet as fructose. The third one, galactose, this is the one that is found, you're going to notice the word lactose, it's a component of milk sugar. So all three of those are monosaccharides, all have the formula C6H12O6. They are hexoses because they are 6 carbons big. The structural formulas look like the linear that you see here, and then they have the same chemical formula listed above, so we call those isomers. Okay? On the next slide, I can just show you, and you don't have to memorize these, so don't spend a lot of time like trying to memorize the structure. I just wanted to bring in a concept that we learned earlier in this unit, which was the word aldehyde and ketone. Do you remember that? Okay, so we talked about with the functional groups, an aldehyde is when the carbon double bonded to oxygen, the carbonyl functional group, is at the terminal carbon. So in glyceraldehyde and in ribose, in glucose and galactose, all the way across, you notice that they are all what we call aldose sugars. Okay, they're oses because they're sugars, and they're aldoses because they are aldehydes due to the carbonyl group located at the terminal position. Okay, now if you look at this subgrouping, we can further subdivide them into triose aldoses. Okay, do you see how we're breaking this up? It's an aldose because the carbonyl is on the terminal position. It's a triose because it's a 3-carbon sugar. That's glyceraldehyde. Okay, we have pentose sugars, 5, that means 5 carbons. It's still an aldose because the carbon is at the terminal position. And then these are the hexose ones, 6 carbons, terminal carbonyl group, therefore those are aldoses. Okay, if you look at the bottom 3 examples down at the bottom, these are ketoses because look at my carbonyl group embedded in the chain. Okay, there you go. And we can further subdivide these. If you go back up top, okay, up here, 3 carbons, so this is a triose ketose. This one over here is a pentose ketose, known as ribulose. And then this one on the very end, which is fructose, is a ketose, but because it's a 6-carbon, it's a hexose. See all the vocabulary? You're using all your root words, your prefixes and suffixes, and putting them together to be able to recognize these structures in biochemistry. Okay, like I said, you don't have to memorize them. I just wanted you to be able to apply what we learned about those functional groups to these different structures. Okay? All right, the next couple of slides should be pretty easy because the hard one was getting the monosaccharides down. So in the next slide, we're going to talk about if we put the prefix di in front of saccharide, now we get 2 sugars. Okay, so mono is 1, di is 2, saccharide is sugar, now we get 2 sugars. Okay, so use what I already taught you, which is the process known as dehydration synthesis. Okay, also called condensation reaction. If we bring any of our 2 monosaccharides together, we join them, it's going to be a covalent bond between the 2 molecules. Okay, so we're going to be sharing valence electrons. And we're going to do this through the process of dehydration synthesis. So what are we going to lose, y'all? Hopefully you said water. Here it is right here. We're going to lose water, and we're going to have a covalent bond formed between them. Okay, the 3 examples that we talk about in biology, these are 3 disaccharides, sucrose, lactose, and maltose. Okay, so all 3 of those, just to let you know, their base is glue, because sucrose is glue plus fruc, okay, F-R-U-C, I'm going to abbreviate. Lactose is glucose plus galactose, G-A-L, galactose, and maltose is glue plus glue. Okay, do you notice how glucose has to be a component of all 3 disaccharides? That just kind of shows you how it's the preferred energy source. We have to have glucose as one of the monosaccharides in all 3 of our disaccharides. Okay, so sucrose is glue and fruc, lactose is glue and galact, maltose is glue and glue. And in all 3 disaccharides, we're going to have a covalent bond between glue and fruc, covalent bond between glue and galact, and covalent bond between glue and glue. So we're going to have sharing of valence electrons between those monosaccharides, okay? So if I look over here in this picture, this is a perfect example of dehydration synthesis in action. Here's a glucose and a glucose shown with the ring structure. Okay, there's the ring structure. And they come close enough where you see here's a hydroxyl group on one and a hydroxyl group on the other. Well, in chemistry, if they come close enough, there's a pull right here where the OH is going to pull the H toward it. As that happens, I get water as a result. So I'm going to knock this off. Knock off the H, joins with this OH to become water. If I knock that off, look what I have left. There's a carbon here and a carbon here that now are not satisfied because they lost a OH here. This carbon is not satisfied. This oxygen is not satisfied because it lost its H. My oxygen still needs one electron to be happy. My carbon still needs one electron to be happy. So there's a bond formed between those two, and you can see it in the picture. There's the oxygen brought down. That's that oxygen. And the bond is now between the carbon there and the carbon at that position. That is my covalent bond, and there is a specific name for it called a glycosidic, this is a D, glycosidic linkage, because it is the specific name of the covalent bond between two monosaccharides, between two monos. So that is another vocabulary term. We call it a glycosidic linkage. It's a covalent bond, but it's the name of the covalent bond that joins between our two monosaccharides. Okay, make sense? All right. Okay, one thing I want to mention before I switch slides, I want to talk about, let's say, lactose for a minute, because I know a lot of you guys have heard of this, and some of you may actually be what we call lactose intolerant. Can you guys think of, like, the biology, the physiology behind that? Like, what would make you lactose intolerant? Can you think of it? Okay, now I can't hear your answers, but hopefully you said something about maybe you are lacking the enzymes. Remember how we already talked about enzymes? If you're lactose intolerant, you're missing the enzymes that are needed to break that covalent bond, to break that glycosidic linkage between glucose and galactose. So if you buy products at the grocery store, and I'm sure you guys have heard of stuff like Lactaid, they sell it right by the milk. Okay, they actually sell Lactaid milk. They also sell Lactaid pills. It's the milk that already has the enzymes added to it so that it's broken down in a simpler form. It's already a monomer form by the time you eat it so that you can actually use it. Because if you can't break down and hydrolyze that bond, or you do it at a slow rate, because that's usually your intolerance, is you do it at a much slower rate, because remember, enzymes only can speed up the rate of a reaction. So if you are missing those enzymes, your rate is much slower. It's going to take you a lot longer to hydrolyze that, so you usually get symptoms like a stomach ache, diarrhea, some gas, etc. And so the intolerance to that is the inability to break down the bond in a timely fashion because your enzymes are not working appropriately. So I wanted to mention that. Okay, one other thing I want to mention before we go on to the polysaccharides is I want to talk about in your book, I don't have a slide here, but in your book it mentions these oligosaccharides. So I want to make sure I mention this really quickly. Oligosaccharides we'll hit on later, but it's several monosaccharides, okay, so ultimately we would call it a polysaccharide, but it's a special group of these, so it's several monosaccharides joined together by the glycosidic linkages, and they have some additional functional groups attached to them. So they're special. They're going to play a really big role in cell identification because they're going to be on the outside of cells. A perfect example is when we talk about ABO blood groups, like how you're identified as type A blood, type B blood, type AB blood, type O blood. When we start to identify those cells, those red blood cells, by their ID tags, these oligosaccharides play a critical role in determining that. So it is an example of several monosaccharides bonded together, so it's bigger than a disaccharide, not quite as big as our polysaccharides, but they do have special properties because they have some additional functional groups added to them, and they play a role in cell identification. Okay, so we'll hit those when we get to the cell and we talk about the cell membrane. Okay, the last thing we need to mention are our polysaccharides. Okay, so this is the last grouping. So if you think of the prefix poly, poly means many. So if I have poly added on to saccharide, I get many sugars big. Okay, now I'm going to go ahead and tell you, we split these up into two main groups of polysaccharides. We have those that are energy storing ones, and on the next slide, we have those that serve a structural purpose. Okay, structure. So we've got energy storage and structural as far as the groupings of those. And same thing as disaccharides, we're going to have dehydration synthesis join many monosaccharides covalently together. Okay, so let's take a look at the two that we have that are energy storing. Okay, I put two little pictures out here. One of these is found in plants and one is found in animals. That's the main difference between them. So starch is the example of the plant energy storing polysaccharide. And they do this whenever they go through photosynthesis and they make a lot of glucose. Now remember, the glucose is made at the leaves. So as they make a lot of it, they store it in little organelles, little structures called plastids. As it builds up, sometimes they send the excess even to the root of the plant, such as a potato plant, where you would get, big tubers form from that. And we do have, look at this, we do have the enzymes that are needed to hydrolyze this into monomers and use it for our energy source. So this is a good one. We eat this a lot. If you ever, eat baked potatoes or French fries or anything mashed potatoes, you're definitely consuming a lot of starch. Same thing even with rice. Okay, these are polysaccharides that have many, many, many monosaccharides bonded together. Now I will tell you, all four of our polysaccharides, all four of them, I'm going to change the color here so you can notice this, all of ours are glucose monomers. That means it's glucose bonded to glucose, bonded to glucose, to glucose, to glucose, to glucose, to glucose. They are all four made up of glucose monomers. What makes them different is how branched they are. Like you see down here, glycogen is highly branched, as well as the number of monosaccharides that is found in each of the polysaccharide chains. Okay, so starch is from plants. We can break it down. We have the enzymes needed. It's the stored form of glucose monomers. Okay, down here, this one is glycogen. Same concept, it's still going to be monomers of glucose. The difference is it's stored up in animals. It's more highly branched. It's found mainly in our liver and muscle cells. And if I actually tie the two together, if you bring these two together, let's say you eat this potato right here. You eat it, you break it down, okay, you hydrolyze it into monomers of glucose, these blue dots, glucose, 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 all these blue dots, and then let's say you use the first two in cell respiration for ATP, but then you've got all these excess blue dots, these are your glucose molecules. You're not just going to have them floating in the blood. You can store them up in your muscle cells and store them up in your liver for later use as this substance called glycogen. So it's kind of like the recycling of glucose. Ultimately, it comes from plants. They store it as starch, and then we can eat it, break it down, and when we have excess, we store it as glycogen. It's like the beautiful working of plants and us together. Okay, the last two, these are my favorites. If you remember from freshman year, I talked a lot about these two. Okay, these are structural. The function of these polysaccharides is a structural component, and here are the two, cellulose and chitin. Okay, now you know I always joked and I said chitin, because I said, are you chitting me? But it's really chitin. Okay, so cellulose and chitin. The first one, cellulose, is also a polymer of glucoses. So once again, it's glucose to glucose to glucose to glucose to glucose. But it's very different because look what it's telling you. Every other glucose is upside down. Look at the picture. You can see it right here. Every other one is in an upside down position. And so that creates a unique parallel strand of glucose that also is held together by hydrogen bonds. So we have parallel strands that are bonded to each other that gives it a little bit stronger than just a single glucose to glucose to glucose. Okay, so watch my dots. If that's glucose, 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 I have another glucose, 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 and these are, I'll change the color to red, hydrogen bonded, okay, hydrogen bonded, hydrogen bonded, hydrogen bonded, hydrogen bonded, and that makes this a more structural basis, which is why it's found in the cell walls of plants. So if you look right here, there's the cell wall. Makes it look like that rigid structure in plants because it's got this polymer of glucoses that are hydrogen bonded together in parallel strands. Okay, the chitin, the last one, is not found in plants. It is found in this ugly looking beetle, It's also found in this cicada down here, because those things have exoskeletons, which are these hard shells, okay, those hard shells, and also look at what it says about fungi. In the cell walls of fungi, which are more rigid, they're going to have the substance called chitin. Okay, so it is definitely stronger because it gives it a structural basis, not a storage basis. Okay? Now the last thing I wanted to mention, this is kind of a little extra bullet here. I'm going to change to a green, paint a green color. If you think about this one, the cellulose, this one is definitely important to us because of the fiber. We do not have the enzymes available to break down cellulose. We literally cannot hydrolyze the cellulose that's in a plant cell wall. So the benefit for us is it goes right through our digestive tract. It helps us have a bowel movement every single day. We call that the fiber. So even though we can't hydrolyze the bonds between those glucose molecules, we lack those enzymes, we still can use it for our fiber in our diet. Amazing, right? Pretty cool. Okay, that concludes our lecture on carbs. Have a great night.